Okay, so today's class is a continuation of the previous two classes, which is getting to know of the various enzymes by which we can manipulate DNA. And in my last class, I had told you about restriction enzymes and the various category of restriction enzymes. And in today's class, we'll be discussing about artificial restriction enzymes and their advantages. So when we talk about the restriction enzymes, if you remember, we were talking about those restriction enzymes which have a specificity, and it's only when they recognize a particular DNA sequence, which in general is around 4 to 8 base pair. They go and bind there and make a cut there with their catalytic activities or nuclease activity. So in that context, since their recognition sequence is limited to 4 to 8 base pair, or yeah, in this range only, and maybe for some enzymes it's little bit longer. If our objective is to make a specific cut in the whole genome for genome editing purposes, and when I'm talking about genome editing purposes, it is that if, for example, I want to remove a particular stretch of DNA in the whole genome, then how do I cut there? I cannot be totally dependent on the fact that the site for restriction enzyme is present there, means which is flanking the region which I want to remove. So I can't be totally dependent on that because it's a chance event. Means I may or may not get the restriction enzyme sites there, and even if I get restriction enzyme sites there, there is a very high chance that those RE sites will be present elsewhere in the genome as well. So somewhere I have to devise a way to make a cut at precise location which is present only once in the whole genome. So that, for precision purpose, I need that particular strategy. So in that context, it was recognized that how about we make the artificial restriction enzymes in the context that we swap their DNA binding domain with the DNA binding domain of certain other proteins, which those proteins are, I just come to those, and then use the nuclease activity of these REs to make a precise cut there. Okay, so in that context, I will be talking about the three strategies for making the cut in the genome, and those are zinc finger nucleases, those are talons, and then there are CRISPR. So among these, I hope that you all know that most famous among these are CRISPRs, which is purely due to the fact of the ease of using CRISPR-Cas9 technology and making a precise cut anywhere in the genome. And but the other two are also not like completely disregarded technologies, and still there are many labs which are actually working on those. So coming to zinc finger nucleases, I've already told you that the idea was that you want to swap the DNA binding domain of the known restriction enzymes and replace it with the DNA binding domain of certain other proteins, and means which can go and bind to a particular region and make a cut with the nuclease activity of the restriction enzymes. So talking about zinc finger nuclease, here let me first talk about how it recognizes a particular sequence of DNA. So zinc finger domain, it comes from the zinc finger transcription factors, if you have heard about those. So transcription factors are the proteins which go and bind to certain region in the genome, and then they promote the transcription of that particular gene wherever they bind, right? So in this context, we just take the zinc finger domain from those transcription factors, and each domain, each finger, it binds to, it's around 30 amino acid, and it binds to or recognizes around 3 to 4 nucleotides. And similarly, so the idea was that if we put together the various fingers, each of which recognize 3 bases, Then let's say if I combine 3 different zinc fingers together to make a particular domain, then it means that I can recognize 9 bases. And if I have more of these fingers, let's say I have 5 fingers, so it means I can precisely recognize 15 bases, right? So this was the idea behind using zinc fingers because of their precise binding to 3 to 4 nucleotides in the DNA. Then the idea was, I told you that to combine it with the nuclease activity. So in that context, they use a restriction endonuclease called FOC1. So FOC1 belongs to type 2S restriction enzyme. And if you remember that the restriction enzymes that I talked about in the last class, they were mainly belonging to type 2 category, the ones which are magnesium requiring, and they make precise cut in their recognition sequence or near their recognition sequence. But in the type 2 category, there are various subcategories which I had briefly told you about. So FOC1 belongs to 2S in the context that it recognizes a particular sequence which is GGATG, and it makes a cut not within that sequence but few bases away from that sequence. Where does it make a cut? So it introduces one cut is made 9 nucleotide away from the recognition sequence, and another cut is made 3 prime and another cut is made 13 nucleotides away from the 5 prime end of the bottom strand sequence. So FOC1 restriction enzyme, it works as a dimer, and what they have done is that they have they have attached one monomer to this particular domain and one monomer to this particular domain, and that increases the specificity. So you can understand that if I have this particular moiety, so this will recognize, let's say, 9 bases here, and then it is means bound to this FOC1 nuclease monomer, but the monomer by itself will not be able to make a cut here, right? So what I do is that I make another moiety which is able to bind somewhere close to the target site, and this again will recognize 9 base. So there is the possibility that in the whole genome these 9 bases are present, let's say at 3-4 sites in the whole genome. But the possibility that these 9 bases are followed by these 9 bases in the whole genome will be very, very rare. So this is from where the precision comes, that you have to closely space these sequences which are present in the genome, and you design it in such a way that they make a cut in between, somewhere in between the two recognition sequences. Now this is also attached to a monomer. Now when these two they bind, then these two monomers they come together, and I told you that FOC1 only works as a nuclease when it is present as a dimer. So once it is now able to dimerize at this particular site, which is our target site, it will make these staggered end cuts. Okay, one 9 nucleotide from the 3 prime end of its recognition on the top strand, and one 13 nucleotide. So it will make a staggered cut. So now in the whole genome, if you want to, let's say, remove a particular stretch of DNA, then you will need 2 sets of these. For example, I want to make, let's say this is whole DNA stretch. I want to remove this particular region. So I will have this particular whole thing that I've designed, and near to that I will have another set which I have designed to make a cut here. So then what will happen? One cut will be made here and one cut will be made here, and this intervening region can be easily removed and this will be rejoined. So how it will be rejoined? By the DNA repair machinery which is present in any cell, and that DNA repair will be either it will be non-homologous end joining, or it could be homologous dependent recombination. So there are two ways by which the DNA ends, they can be repaired, and I will briefly mention it towards the end of the lecture as well. So this is in all about zinc finger nucleases, how they can make a precise cut in the genomic DNA. So any questions with respect to this? Okay, so another thing is that designing of these zinc finger nucleases is comparatively difficult, because now you can understand that there are 4 bases in the total which are known, A, T, G, C, and I'm saying that each finger in general it recognizes around 3 nucleotides. So it means that you have to design these fingers in such a way that they recognize a particular 3 base stretch. It could be GGG, it could be GCG, it could be triple T, it could be triple A. So means there are so many permutation combination. So it means 64 combinations it has to recognize. So it means we need 64 different zinc fingers which have the capacity to bind to any of these combinations that I'm talking about, and with the precision in such a way that it only recognizes, let's say, triple G. It does not recognize GCG. Means it should not be tolerant of even a single base pair mismatch, because when we are talking about genome editing, we are talking about something which is extremely precise in nature, and obviously you do not want to alter anything else in the genome of any organism, but only a particular stretch that you are interested in. So off target effects are something that are of very prime importance when we are talking in terms of genome editing. So be it for ZFNs or talents or CRISPR, off target effects are something that need to be taken care of, and still scientists are trying to understand that what guides the interaction between each finger and the 3 bases or few bases that I'm talking about, and what all, under what conditions means this may be affected, and how much tolerant it is to the mismatches, etc. So that's why designing these is very, very difficult. And I know that Professor Sundar from our department, he used to work on ZFNs before, and so he's a bioinformatics person, so he would design ZFNs for precise locations. So he made some, I think, softwares and databases also for the same. And now he's working on CRISPR and all. And I think that the first person to talk to, if you're very interested in any of these things, would be Professor Sundar in our department. So he's into designing of ZFNs or CRISPR for precise editing, and also understanding that what interactions, whether the water content or these means which interactions, and I don't know, the various parameters they work about, hydrophobic interactions and so on, which affect the DNA and protein binding, etc. So coming to the next strategy, which is talents, they are considered comparatively easier to work with as compared to ZFNs, because in ZFNs you have to design each finger which will bind to a triplet. And in case of talents, each moiety here, each tail recognizes 1 base. Okay. So what are talents? These are transcription activator like effector tail, which are combined to the nucleases, and in this case also the nuclease that we are talking about is FOC1 only. So here also exactly same strategy is being used, but only difference is the recognition of these tails with the these recognition, this DNA sequence. Actually, I'm just remembering, is this lecture being recorded?
Yes, ma'am.
Okay, okay. Thank you, thank you. I just got confused whether I clicked that thing or not. Okay, so yeah, so that is the only difference between talents and ZFNs. Now coming to what tails were. So tails, these were actually recognized that these were the proteins which were injected into certain bacteria. Sorry, these were injected by certain bacteria into plant cells. So these were the proteins, and these proteins, they will go and bind to certain DNA sequences in the plant cell throughout the genome. And these proteins, because they were transcription activators, so not only do they have some recognition property, but they act as activators of the transcription. So they will lead to induction in transcription of certain genes which are present in the plants, which will help the bacteria in some way. Okay, so in short, that is the introduction to how this battery of proteins were initially recognized. Okay, so when the molecular biologists, they try to understand what is the basis of interaction between tails and the DNA sequence, they realize that each tail, each of this box that I'm showing you, it is like 34 amino acid. This recognizes one base. Okay, so now if I say that I want to, let's say, recognize 18 bases on this side and 18 bases on this side, it means I have to add 18 tails together, and at the 3' end of which will be the fork1 moiety. Similarly, on this side also, because fork1 works only as a dimer, anyway, I would need a recognition on this side also. So again, I will have 18 tails which will be joined together, and on the end will be the fork1 nuclease activity, and in between it will make a precise cut. So this is how tails work. And because here you don't have to, really worry about the fact that what will be the combination of nucleotides here. So because for each nucleotide you have a tail which is ready, and this tail means scientists have worked about that it is extremely precise in nature. It will not bind to the other nucleotide. So it is just you have to put together all these things, and then it will make a double strand cut. Once a double strand cut is made, then it can be rejoined by the DNA repair machinery. So this is about talents, and I told you talents are comparatively easier to work as compared to ZFNs. And there were some observations, some papers which have been published in which it was shown that talents were more precise than ZFNs. And also their genome editing efficiency was higher than ZFNs. Okay. Now coming to the CRISPR-Cas9 based technology. So before I come to this, I also want to tell you that for each of the slides that I've shown here, there are notes, the details which are given, which are in the notes section, or the links are already provided in the notes section. Okay, so CRISPR-Cas technology, I hope that you all know that these two scientists, they got the Nobel Prize for the same. And the Nobel Prize was given precisely due to the fact because of the ease of this technology and the immense potential of this technology in performing genome editing, and the final potential not only in functional genomics, in which we want to, for basic understanding of the functioning of a particular stretch of DNA, it could be gene function, etc. But also that its potential to actually change the means doing editing in the genome and correcting certain diseases, or adding some useful character in plants or bacteria or yeast, which will be very, very useful for biotech industry or in the health industry. So coming to these CRISPRs, so let's understand what's the full form of CRISPR. So CRISPR means clustered, regularly interspaced, short palindromic repeats. Okay. So when I'm talking about repeats, you please see these black lines which are interspersed between these green boxes. I'm talking about these repeats. So these are regularly interspaced and clustered repeats which are present in the genome of various prokaryotes. Okay, so these are called as CRISPR loci. So black lines are the repeats, and the green boxes, what these are? These are the remnants of the previous infection in that bacteria by certain bacteriophage or some foreign DNA. It could be plasmid DNA, etc. So when these bacteria, they were infected by various let's say bacteriophage, or some foreign DNA had come in their within the bacteria, you know that it is generally acted upon by various restriction enzymes, and it is cleaved into small pieces, right? That's how I told you that restriction modification system in my last class, that how bacteria restricts the entry of the foreign DNA and protects itself. So it makes the cut in the foreign DNA by restriction enzymes. But at the same time, it was seen that some of these fragments which were created, they get incorporated into the bacterial genome. So if we see the CRISPR loci in the bacterial genome, we can see that let's say if this region is there, and it is part of some foreign DNA previous infection by some foreign bacteria. Okay, so here all these colors are same color, suggesting that these all remnants are from the same bacteria, but that's not actually the case. You can consider these all differently colored, means this can be from some other infection, this can be from some other infection, and so on. Okay, so this is the CRISPR loci, which consists of spacers as well as repeats. So these green boxes, they are called as spacers. Then there is a leader region, and then there is a battery of Cas genes, which is nothing but CRISPR associated genes. And these genes, they have varying functions. So these proteins, they were shown to have nuclease activity, which we actually require when I'm talking about CRISPR based genome editing. But some of these, they also have certain modification activities, or even transcriptional promoter activities, etc. But when we are talking about genome editing, we'll be talking about only those Cas genes which do have nuclease activity. Okay, so the CRISPR loci includes Cas genes, a leader sequence, and several spacer sequences derived from engineered or foreign DNA that are separated by short repeat sequences, and individual mature CRISPR. So this gives rise to CRISPR RNA, and this then gets processed into small CRISPR RNA. So I can say that in a way this is like primary or unprocessed transcript, which on processing gives very small these CRISPR RNAs. These CRISPR RNAs, then they get associated with certain transactivating RNAs called tra-RNAs, and these two together then go and bind to the target sequence and recruit the Cas9 gene here, and then makes a precise cut. Okay. Now, in normal circumstances, I mean, when I'm talking about immunity of the prokaryote to any further infection by the previous bacteriophage, what it does is, let's say there is a fresh bacteriophage infection, the remnant of which is present here. So this remnant means whenever the infection is there, it will give rise to CRISPR RNA, and this RNA will then go and bind to bacteriophage DNA and cut it. So as a result, in a way, it is like the bacteria has now got immune to the infection with that particular bacteriophage. Now coming to that, how do we design the CRISPR for making precise cut in our genomic DNA at the precise location that we are interested in? So what we do is that we design a short hairpin RNA for a region that is of interest to us. Now, what is short hairpin RNA? So it is simply in, sorry, not short hairpin, it is short guide RNA, sgRNA. Actually, I'll show you this slide. See, I had planned to show you a very good video for CRISPR, and but I realized that the volume is not high enough for you to listen to it. So I've given the video links for each of these things, and for CRISPR especially it is very well presented here. Okay, but from my side, let me try to explain that how do you do the cut at precise location using CRISPR-Cas technology. So if you are asked to make a cut at a particular region, what you will do is that you will design a RNA which is complementary to it. Okay, only thing is that you have to design it at such a location that it is followed by three base pair. So this is called as protospacer adjacent motif. So it was seen that Cas9 will be only able to cut at those locations in which the region is followed by a sequence called PAM. Now this PAM, it varies with the various Cas proteins. There is Cas 1, 2, 3, 4, 5, 9 and so on, right? So for Cas9 protein, which is generally used, its PAM sequence, this PAM motif is NGT. So you have to design it from such a region in which this will be, uh, complementary to the region of your interest where you want to make a cut, but it should be followed by NGT. That should be the only consideration when you have to design a sgRNA. So, and do you think it is a like very big problem in finding such region? Maybe not, because PAM region is nothing but NGG. So finding this region is, I understand that yes, it comes as a means one caveat, but still it's not that bigger region that is like completely impossible to find. So usually people are able to find region which in which sgRNA is followed by NGG. So in that case, what is sgRNA? I already told you, it is like CRISPR RNA, crRNA, which I had shown you in my previous slide. And previously, this would have to be combined with TRA RNA, that trans-activating RNA. But in this case, in the vectors itself, the TRA RNA coding region is there. So what you get finally from your vector is sgRNA getting synthesized. So once this sgRNA, you put this vector inside, which has gene coding for sgRNA and a gene coding for Cas9. So what will happen? This sgRNA will go and bind to your gene of interest, and then because it has PAM motif next to it, it will Cas will recognize it, and it will make precisely a cut 3 base pair before the PAM region. So here the cut will be made, and now you have a very clear double-stranded cut in your genome. You can understand that the designing of sgRNA is so much easier as compared to designing of ZFNs and talents. It is not dependent on that you have to actually identify that which finger will bind where. It is simply complementarity. If you have a region GGATTC or whatever the region is, you just have to design a complementary region. That's it. So, and the editing is also extremely simple. You just have to put a vector inside your plant cell or a human cell or a bacterial cell, and it will go and recognize that region and make a cut there. Then how do you identify the cells where the cut has been made is a different story, and how efficient it is another different story, etc. But the possibility to make a cut at any sequence in the genome is what is the strength of CRISPR-Cas9. So in case of ZFNs, I've told you each finger recognizes around 3 base pair. Here talents, each module recognizes 1 base pair, and in CRISPR-Cas, 1 base is recognized, means it's one base pair each, right? So these are the three major technologies, and the efficiency is highly dependent on that how good is the DNA repair efficiency of the organism you are working upon. So because the DNA cut is made, and let's say two cuts are made in the genome, now you want to remove the intervening region. So you're totally dependent on the cell to combine these two regions, and this will be gone. So from there the efficiency comes, that how efficient your cell is in making the repair of that particular thing, and that too in precise repair I'm talking about. Apart from these three technologies, there is something else that many companies or scientists they are working on. It is called meganucleases. So they are simply, I would say, restriction enzymes, but with extremely big recognition sequence, very big. So it ranges from more than 12, actually in some books it's written more than 12, in some like 20 to 40 base pair, but I would say yes, more than 12 to up to 40 base pair is its recognition sequence. So if we have some enzyme which can recognize these 40 base pair or 30 base pair, so it means I have extreme specificity in my whole genome, which I was actually requiring. So that's why many people are now working on meganucleases, and these meganucleases they are able to make a cut, a double stranded cut, just like any of these enzymes. And how they are, see, there are some naturally occurring meganucleases which will recognize a particular sequence, but now I'm talking about the sequence which I want, which can be any sequence stretch in the 3 billion base pair in humans or in other organisms. So how do I get that precision? So what they are doing is they are trying to make, first understand that what guides the recognition by this meganuclease of these DNA sequence, because unless you have a basic understanding of that, you cannot alter the recognition capabilities. And then what they are doing is they are doing, once they have identified that what is the motif that recognizes the DNA sequence, they are making random mutation in those motifs and trying to change the DNA recognition ability of these meganucleases. So this is what they are doing. But if means if I talk about myself, I find it comparatively not user friendly, because for a particular stretch, how many mutations and how many changes will you make in a meganuclease to actually make it cut at a precise location? So if I talk about any of these things and the utility involved, definitely CRISPR-Cas is the most easy technology as compared to any of these three or four. So that is why many labs have already incorporated CRISPR-Cas9 technology in their respective labs. Just for your information, I would also like to tell you that it's not only CRISPR-Cas9,
Ma'am, your voice is not coming.
Okay, can you see the screen? No. Can you see?
No, ma'am.
Yes, ma'am.
Okay. It's already recording. Okay. Yes, so that's all about the four different technologies. And what I wanted to tell you is that you please see all these videos which are given here, and also you can please see this ethical concerns which are associated with CRISPR-Cas9 technology. And there is one article that has been there are several articles. You must have heard about the children, the embryo editing of two girls in China, and how that scientist was put under. So he's in jail right now. So he did not obviously take the this consent, and he had edited two unborn children, and those two girls are born now. And are you aware of this case? Any of you?
Yes, ma'am.
Yes, ma'am.
Okay. Yeah, so you please read about this, because see, you are editing a genome, and your intentions might be fine. One point is that, okay, you want to correct a particular disease, but obviously you have to make anybody who is having this editing done to be aware of the plus and minus of the same. First point is that, are you really sure that your CRISPR-Cas9 or ZFN, whatever you are doing, does it make a precise cut or not? Because there are now many papers which are coming that they found off-target effects of CRISPR-Cas9 as well. So the scientist I'm talking about, he had edited a gene in the at the embryo stage in the genome, which codes for CCR5. And this CCR5 is a gene. It is a receptor for HIV. So HIV, this virus, it binds to CCR5 and then makes entry into the cell. So there was one observation in certain populations that those individuals who have mutations or deletions in CCR5, they are comparatively resistant to HIV infection. So, because the father of these babies, he was HIV positive and mother was HIV negative, so he thought that he has to make the babies who are not, who are, let's say, completely resistant to HIV infection. So he did in vitro fertilization, and at the embryo stage, he edited the genome, and the CCR5 gene was mutated. So which type of mutation did he introduce by CRISPR-Cas9 technology? Was a frame shift mutation. So as a result, CCR5 is not functional, because in a frame shift, the protein was not being made at all, hardly. So as a result, CCR5, because it is not there, so likely the girls who are born, they will always be resistant to HIV infection. But then, can he ultimately ensure that? Are all HIV viruses, they are completely dependent on CCR5? Can he ensure that the CCR5 gene that he has edited is not involved in any other function in the cell? um, Maybe it is involved in other functions. You now know that each protein has several interacting proteins and is involved in multiple pathways. So we don't know, by editing one gene, maybe we are making them resistant to HIV, but maybe we are making certain other changes which are not desired. So we don't know. So that's why ethical concerns with respect to genome editing is a very, very big thing. And now the scientific community is taking a big note of the same. And I already told you that scientist is now under, he's serving in jail. And also many other scientists are being questioned, who he had discussed before doing the editing that he's planning to do so. So why they were not so aggressively, they ensured that he did not do this experiment, which is not ethically right. So means please go through this article, and there are many articles who are for you to actually read. Uh, then one last slide that I would like to show to you is that generally when you make changes, when you do you perform double strand breaks by ZFN or Talens or CRISPR, not shown here. I couldn't find the appropriate slide. Then what could be the final consequences? I told you that these breaks, they get repaired by DNA repair machinery by any of NHEJ or HDR dependent pathway. Now the point is that if I want to remove a particular stretch of DNA, what I will do, I will make two precise cuts, right? One here and maybe one here. And then the middle region will be removed, and I'm assuming that the repair will take place, and the two, let's say now in this case, let's say a chromosome is divided into two halves, they will come back together, right? Now, if I want to replace let's say that here there was a mutation and I want to correct it. So in that context, what I will do, that I will make a cut here and I will make a cut here, and I can give a template DNA inside my cell. So that template DNA will have the corrected nucleotide, and through homology dependent recombination, my corrected DNA will be incorporated into the genome. So in that context, what I am doing, I am not removing any, a stretch of DNA. I am just replacing the defective DNA with the correct DNA by providing the correct DNA as the template DNA through homology dependent recombination. Can there be some other things that I can do? Yes, I can. Obviously, you can delete. You can perform inversion also by providing a stretch of DNA, or incorporate removing one stretch of DNA and incorporating it in the inverse orientation. Or what you can do, you can also perform translocation. So in translocation, what happens? Translocation is when you have two stretches of DNA which are present at elsewhere in the genome, means at two different locations coming together. So this translocation can be within one chromosome, that let's say two stretches of DNA which are present on two ends of the chromosome, we are able to bring them together. Or it could be that translocation in trans. I mean that the two different chromosome fragments, they come together and they make a completely different stretch of DNA. So all these things are theoretically possible. Have they been done yet? I'm not sure about, at least translocation I have not heard yet. Maybe there are some experiments going on, but you can understand in case of translocation, what you have to do is you have to make cuts at two different chromosomal locations. You have to get two different fragments, and you have to depend upon the chance event within the cell of repair taking place, in which the two stretches of DNA which are now, let's say, floating in the cell of coming together, and their DNA being repaired, means the ligation taking place of these two DNA fragments and making a completely new stretch of DNA. So when I'm talking about deletion, translocation, etc., they are important when you want to understand that what is, for example, a role of a particular translocation, which is generally seen in various patients, or what if means from means functional point of view, if you want to understand the importance of a particular stretch of DNA, then you can do all those things. But for corrective thing, generally what you do is that you supply with a new stretch of DNA, or let's say if there is some extra base which is there and that base is causing a particular disease, then you can remove a particular one base of that particular extra base in the DNA. Or in some cases, you can add, let's say if a gene is deleted, you can add the gene by providing the gene from outside and at a precise location. That is what I want to say. So that is all from my side, and you can please go through the various links that I have given here, and now I'm open to questions.
Ma'am, I had a question. In cases of zinc finger nucleases and talents,
Hmm.
so ultimately we are also restricted by the recognition sites of the restriction endonucleases. Ma'am, is it
No, no,
possible?
not okay. Okay, wait. For restriction endonuclease, you don't use their recognition sequence that has been removed. So FOC1 just has the catalytic domain in this case. Its own recognition sequence, which was I think GGA, TGG, or whatever it's given in the slide. That motif has been removed from FOC1. It just has the nuclease activity in this case.
Oh, okay. So any sort of endonuclease can be also used in the cases as well, instead of just
Right. We need
just.
the nuclease which makes a cut not at the recognition sequence but close to it. It depends that what sort of thing. See, in this case, see, if I want to remove a particular stretch of DNA, what I do is I design ZFNs which are flanking that stretch of DNA, right? If I had used a nuclease activity which makes a cut at the recognition sequence, then the cut would have been made in exactly the same position. But in this case, I want to remove a particular stretch of DNA. So in general, FOC1 is used which makes a cut near the recognition sequence.
Okay, ma'am. Thank you, ma'am.
Okay, so any more questions?